So welcome everyone to our Acta STEM presentation today. So as part of Acta STEM, the registries aspect has recently joined, and the idea has been to integrate statisticians working in registries and any people working in clinical research in registries as well into the sort of the Acta STEM grouping, and so sort of merging the statisticians of the sort of the clinical trials and registries disciplines, because there's a lot in common. So my name is Andrew Fords from Monash University, and I co-chair the Acta STEM group with Kate Lee, who is unfortunately apologies today. So very pleased today to be presenting. I think it's one of our first registry content sessions, and this is with Errol Ernest and Jessie Hanson. But first, I'd like to do an acknowledgement of country. I'd like to begin by acknowledging the traditional custodians on the lands which I am here today, which is the Bunurong people of the Kulin Nations, and of the people of the nations where all of you are located right at the moment, and pay respects to their elders, past, present, and emerging. So a little bit of housekeeping. Thanks Ben for driving the slides. Please stay on mute to ask questions. Use the chat function in Teams, which should be at the top toolbar on my screen anyway. And so just use the chat rather than the Q&A function, and I'll keep monitoring the chat. At the end of the session, we'll have time to answer all the questions, but I'll be monitoring the chat throughout. And if anything comes up that's particularly relevant at a particular time, I'll consider whether to sort of interrupt the speaker a bit and sort of ask that question for you if relevant to the content at the time. There's going to be a recording of this webinar that's going to be available, and at the end of the webinar, there's going to be a post-webinar survey. So next slide, please. Okay, I'll let Errol talk about the introduction about benchmarking, but here's a little brief summary. So next slide. Okay, so it's my pleasure to be introducing our two speakers today. They're going to share this session. So Professor Errol Ernest is the Deputy Head of the Clinical Outcomes Data and Reporting and Research Program at School of Public Health and Preventive Medicine, Monash University, and he's Professor of Biostatistics there. He's authored a textbook on how to consult with a statistician, written book chapters and hundreds of publications. His research interest is in Bayesian spatiotemporal models and also machine learning techniques applied to registries. And he's a leading authority on benchmarking in clinical quality registries, which we're going to hear about today. And also Dr. Jessie Hanson, a senior scientist, recently completed a PhD at Monash, which is now the Institute of Medical Informatics. That's a long title. Statistics and Documentation at the Medical University of Graz in Austria. So her PhD was about the benchmarking in clinical registries. So I won't take up any more time with the introductions. You can read all the extra little bits there if you like. And I'll hand over to you, Errol, starting off. Is that correct?
Thank you, Andrew. Thank you very much for the very nice introduction. Actually, I mean, today's presentation is all about Jessie's work, so I will not steal her thunder. What I'm going to do is to actually pass over to Jessie and let Jessie present, and then we'll have a discussion after the presentation. Over to you, Jessie.
Thanks, Arul. I'll just share my slides. Okay. Great. So thank you very much for the opportunity to speak today about the statistical considerations around benchmarking outliers within clinical quality registries. So a bit about my experience and background. I studied epidemiology at the University of Queensland before moving to Melbourne in 2022 to work as a data analyst for clinical quality registries at Monash. During my time there, I was involved with several registries, including the Australian Breast Device Registry, the Australasian Pelvic Floor Procedure Registry, and the Australian-New Zealand Trauma Registry. And during this time, I got asked a lot of questions about benchmarking, when to start, what methods to use, when it's appropriate, when it's not. And I found that there was a lack of evidence around methods and guidelines for implementation of benchmarking within the context of registries. And that's what spurred me to start my PhD on this topic under the supervision of Arul and Susanna Ahern. So I finished this last year and then I moved to Austria, where I now work as a senior scientist at the Institute of Medical Informatics, Statistics and Documentation in Graz. So here is a brief overview of my presentation, which will follow a fairly traditional structure. I'll give a brief background and then present the methods and results from two studies of my doctoral project, a simulation and an empirical study. And then I will run through the key considerations and recommendations that I developed informed by these studies. Afterwards, Aurora will lead a group discussion and a Q&A and a poll. So I'm going to assume that everyone is familiar with registries, so I will dive right into benchmarking. Benchmarking is often conducted to monitor healthcare quality within registries, and it involves the comparison of health provider performance against a benchmark, often the population average. Most commonly, it is applied at the site level, but it can also be used to compare clinicians or jurisdictions. For my work and this talk, I will focus on the comparison of sites. Quality indicators used to assess performance can include processes like diagnostic test completion for monitoring adherence to standard care guidelines, as well as outcomes like mortality and patient reported measures. To account for factors outside of the control of the sites, risk adjustment is often used, and then methods are applied to identify outliers by comparing individual site performance to the benchmark, most commonly with control limits or confidence intervals. And these are often visualized using funnel plots or caterpillar plots, as seen on the slide here. Sites flagged as underperforming outliers can then be investigated by the registry and targeted for performance improvement. But beyond the internal investigation of outliers, there are broader implications. There's interest in the public reporting of benchmarking to inform patients and stakeholders of performance. And in the US and UK, the results of benchmarking are also used for pay-for-performance schemes. So given this, it is vital that appropriate and accurate methods are used. However, the optimal methods and registry conditions for outlier identification within the context of registries were unclear. So to address this gap, I conducted a simulation and empirical study, which I will describe now. So I designed the simulation study to follow the ADEMP framework described by Morris, White, and Crowther, which involves clearly defining the aims, data generating mechanisms, estimate or target, methods, and performance measures for the simulation. I developed a function in Stata to simulate the data sets, apply the methods of interest, and store the performance measures. So this function is open source and available on the Monash Bridges repository. So let me or Aurora know if you'd like a link. I'm very happy for it to be used for other purposes. So the aims of my study were to compare the current and common methods of outlier detection and evaluate their performance. And the target was outlier classification, specifically the categorization of poor or underperforming sites. So using the function in Stata, I parametrically simulated hierarchical clinical registry datasets with 3 levels: patients nested within clinicians, nested within sites, with a known number of true outlier sites using the logit model shown here to randomly generate individual patient-level risk and binary outcomes. The table outlines the registry parameters and the values evaluated in this study. which were indicated prevalence, dispersion, or between site variance, outlier definition, a risk quotient representing the difference in underlying prevalence between non-outlier and outlier sites, clinician to site variance ratio, risk adjustment fit, number of sites, clinicians, and patients, and the case volume minimum, which is the number of patients a site is required to have to be included within the analysis. So the parameter values were chosen based on a systematic review that I conducted to reflect both common values and test more extreme scenarios. And they were varied in a partial factorial framework with a default value chosen for each simulation in which that specific parameter wasn't being varied. And all parameter combinations were evaluated for 3 prevalence levels, 5, 40, and 90%, representing low, mid, and high values. And for each unique parameter combination or scenario, 1,000 simulated data sets were generated. So for the methods, 4 models and 5 outlier classification techniques were selected based on my review and the broader literature, giving a total of 20 model method combinations. The 4 models were unadjusted site outcome rates, in addition to risk-adjusted estimates from 3 logistic regression models, ordinary fixed effects, and random intercept effects. For the outlier classification techniques, 3 control limits, exact binomial, the world approximation to the normal distribution, and the false discovery rate, and 2 confidence interval methods, exact Poisson and the Bayer approximation, were evaluated. And of these, the exact binomial and Bayer method were the best performing. So I will focus on these when presenting the rest of my results. And the statistical performance measures are listed here and included the receiver operator curve, area under the curve, as a summary measure. So for my empirical study, I used data from the Australian New Zealand Society of Cardiothoracic Surgeons, or ANSCITS, their cardiac surgery database, which is a CQR that follows patient care and outcomes after cardiac surgery with 2 key clinical quality indicators, operative mortality and new renal insufficiency. The registry has over 50 sites contributing data and captured almost 40,000 cases between 2018 and 2022. They conduct quarterly site benchmarking of their key outcomes internally and also publish de-identified site benchmarking in their yearly annual report. So using data from their 2022 annual report, I applied benchmarking methods to compare the real-world outlier flagging agreement between approximation and exact methods, confidence intervals and control limits for 2 different significance levels, unadjusted and risk-adjusted outcomes, and rolling or a single year of data and cumulative multi-year data sets. I also conducted a small simulation study to assess the theoretical results between the comparisons. So to the results. So this figure plots the parameter of prevalence on the x-axis against the average simulated AUC performance on the y-axis. Marker and line color differentiate the 8 different model and method combinations presented here. So as we can see, for almost all of the methods, apart from those with random effects estimates in the red, there's a sort of inverse U association between AUC and prevalence, with performance being very poor at very low and very high prevalence due to floor ceiling effects. So random effects was consistently the worst performing method, as the random intercept overshrunk the estimates and removed all between site variation, resulting in few to no outliers being flagged. Fixed effects perform similarly, which is in the purple here, perform similarly, albeit slightly worse, to ordinary logistic regression in the blue. But some fixed effects had some issues with convergence and performance in data scenarios with high prevalence. So benchmarking with unadjusted estimates in the green had consistently lower accuracy than when site rates were adjusted with logistic regression. And results were generally quite similar between control limits and confidence intervals, but control limits performed better in some scenarios, again at high prevalence. So in the interest of time, I will just very quickly run through the results for a selection of the registry parameters. So with increasing dispersion between site variants, there was a reduction in AUC. With increasing risk quotient, there was an increase in AUC for low and mid prevalence. There was little to no association between the number of sites and clinicians with accuracy, but looking at the number of patients per site, there was an increase with increasing patients. And then looking further into that at outcome events per site, there was an increase in AUC until around 100 events per site, after which accuracy plateaued. And with a case volume minimum, AUC was reduced, but the magnitude of the reduction is proportional to the percentage of sites excluded from the analysis with the given minimum. So this figure here gives a visual representation of the differences in real-world outlier flagging for renal insufficiency in the ANSCIDS database. Each figure shows the site case volume against the outcome rate for different adjustment and timeframes. Site flagging by the 4 methods is shown with different markers, and the population average again is marked with a line. So the 2 rows show the different adjustment, unadjusted on top and risk adjusted on the bottom, and the columns show the different time frames, so 2022 on the left and 2018 to 2021 on the right. Looking at each individual graph, you can see that there are more sites with purple, indicating the slightly higher flagging rate with control limits for this data set. And you can see that both the approximation and exact methods flagged the exact same sites with the outside square or diamond and the cross. Comparing the rows, you can see that there are some differences in flagging between unadjusted and risk-adjusted outcomes, although some sites are consistent. And although it isn't possible to directly compare the timeframes using this figure, I know from looking at the site IDs that they are mostly different between the different timeframes. So this table here gives an overview of the empirical and simulation studies for the 4 comparisons. So for the ANSCIDS database, the approximation and exact methods had almost identical outlier flagging and simulated accuracy for all scenarios. There was generally good agreement and similar expected accuracy between flagging using confidence interval and control limit methods, but control limits did perform better for certain scenarios. As expected, 95% intervals and limits flagged more outliers than 99.8%, but there was also a performance trade-off. So 95% limits had a higher sensitivity and 99.8% limits had a higher specificity. Agreement between outliers flagged with unadjusted and risk-adjusted data varied widely, but risk adjustment had consistently better expected accuracy. And outliers flagged using data from a single year were very different to those found when analysing cumulative data. And due to the higher number of patients per site with cumulative data, the expected accuracy was higher than for rolling data. Okay, so to the final section of my talk. So to help consolidate the results from the simulation studies, my final doctoral project was to develop guidelines for implementing the optimal methods to detect site-level outliers in clinical quality registries. So in the guidelines, I developed recommendations for 3 important areas, which I'll go into more detail about next. But these are methodological choices, clinical quality indicator characteristics, and registry size considerations. So in addition to the guidelines and recommendations, using the simulated data, I also developed a prediction models for determining the expected accuracy of benchmarking, and created a dashboard in R-Shiny to help with the implementation of the guidelines and recommendations. So I will now discuss the recommendations in more detail. The first recommendations relate to methodological choices when benchmarking with 3 key points, so risk adjustment and regression model. Where possible, patient outcomes should be risk adjusted to account for non-site sources of variation, and logistic regression should be used for this. To add though, for process indicators, risk adjustment is often not necessary. Outlier classification technique, the method used to flag outliers. So funnel plot control limits using the exact binomial method are recommended for flagging outliers. Although the Wold approximation did perform well in the empirical study, the exact binomial method had better simulated accuracy in instances of extremely low and high prevalence without a high computation cost. And as such, it is the broad recommendation. And although control limits performed very similarly to confidence interval methods, they did perform better in some scenarios and can also be visualized on funnel plots, which have been found to be an appropriate and easily interpretable tool for displaying the results of benchmarking. So the choice of control limit level depends on the audience of reporting and the severity of the indicator. 95% confidence intervals have a higher sensitivity and fewer false negatives at the expense of more false positives, while 99.8% limits have a higher specificity and fewer false positives, but also more false negatives. So given this, 95% limits would be more appropriate for severe outcomes like mortality, where missing underperformers has a high cost, whereas 99.8% limits may be better for public reporting to reduce the risk of reputational damage to sites potentially falsely flagged. And for the case volume minimum, case volume minimums for site inclusion and benchmarking should only be used when there are high numbers of patients per site within a registry, where only a small proportion of sites will be excluded from benchmarking. So the next recommendations relate to clinical quality indicator characteristics, again with 3 main suggestions. So indicator prevalence. When selecting quality indicators for benchmarking, preference indicators with a moderate prevalence, indicators of low, under 10%, and very high, over 95% prevalence, in particular, have quite poor accuracy when used for benchmarking. So for dispersion between site prevalence, preference indicators of low dispersion, but risk adjustment can help to improve this if it's an issue. High between site variance makes it difficult to identify underperformers because there's a lot more noise. Clinical significance. Be mindful of the clinical significance of any statistically significant difference. Larger differences in prevalence between flagged outliers and the benchmark have higher accuracy. Outliers flagged with a small clinically insignificant difference to the benchmark should be carefully considered and may not require further follow-up. And the final recommendation relates to registry size, specifically the number of patients per site. So benchmarking analyses for the purpose of site outlier identification should only commence once a registry has reached a sufficient average number of patients per site. The recommended number of patients varies by prevalence, and a threshold of about 100 events per site is needed to reach peak accuracy. So using this, a registry with an indicator prevalence of 10% would need an average of 1,000 patients per site. But this threshold is for peak accuracy, so over 90% AUC, but benchmarking can be conducted with fewer patients per site, still with high accuracy. So here is a screenshot of the dashboard that I developed. On the left is the input section where a registry can enter their data parameters. So this example is for a registry with a prevalence of 10%, indicated prevalence of 10%, between site standard deviation of 5%, 2,000 patients, 100 clinicians, 20 sites, no case volume minimum, satisfactory risk adjustment fit, a 2-time baseline clinical significance using 95% control limits. On the right of the dashboard are the accuracy predictions. So the first box displays the expected outlier classification performance, giving the predicted sensitivity, specificity, and summary measure of AUC with 95% confidence intervals. The insights and recommendations box gives an overall assessment of the expected accuracy as being poor, acceptable, or high, and provides targeted recommendations relevant to the registry scenario for improving accuracy if given a poor or acceptable score. So for this input, outlier flagging accuracy is predicted to be acceptable, and the recommendation around preferencing outcomes with lower dispersion is shown. Of course, not all of the recommendations are feasible. You can't always choose the dispersion for the specific indicators. Sometimes it is what it is, and further, these predictions are meant as a guide only. The goal is to make sure that those implementing benchmarking are aware of the uncertainty involved with such analysis, particularly under conditions, such as high dispersion, high prevalence. So to end, I would like to emphasize that registries should consider their own setting when deciding when and how to implement benchmarking. It is also really important to consider the purpose and audience of benchmarking, as well as the type and severity of the indicator. Underlying reasons for outlier status should be investigated before naming and shaming or public reporting, including data quality, the patient population, and site resourcing. And finally, further areas for future research include methods to handle benchmark uncertainty and bias, the incorporation of clinical significance into flagging, and the long-term impact of outlier flagging on patient outcomes. So for those interested, here are my relevant publications and a link to the dashboard, which I can also put in the chat. And I'm happy to share these references and slides with everyone. And then I would just like to thank my supervisors and my research groups at Monash for their support and involvement in this work. And I will now hand over to Arul for the group discussion.
Thank you very much, Jessie. What a wonderful presentation. Okay, I'm sure the audience has a couple of questions, but before we get to the questions, if you allow me, I just want to put up a short poll for us to go through a couple of questions, and I'm hoping that those questions will help to shape our discussion. Okay, so I'm just going to copy a link and put it on the chat box. Don't worry, the link doesn't bring you to any spam or any phishing website. It's a link to a Microsoft online forum, right, of online poll. And your responses are anonymous, so don't worry, your name will not be flagged. So let me just share my screen. I'll show you what it looks like. I've just started to share my screen. You should be able to see it shortly. Okay, if you can see my screen, can you just give me a thumbs up? Right, a thumbs up if you can see my screen. Yep, excellent. Okay, so these are a couple of questions. I'm going to just paste the link on the chat box. So I'm going to copy the link. So the questions are more about what kind of challenges we face when we do outlier detection, some of the challenges in developing and disseminating the site reports and the annual reports that we do. And then basically a question about, what kind of topics you're interested for subsequent webinars. So I'm going to copy that link now. I'm going to stop sharing and I'm going to paste it on the chat box. Okay, so I've just pasted it. Now we'll take a few minutes to just go through the response. I will share my screen and then we will look at the general responses. Okay, just click on the link and then go through those few questions. Okay, I'm just reading the chat box. I think there's a comment from Deborah. Thank you, Deborah. Great talk. Could you also share the link to the dashboard and guidelines? Um, so after the presentation, we'll get the actor team. Bernie maybe can help to actually disseminate the link to the dashboard and the presentations themselves. Okay, I'm going to start sharing my screen. Let's look at the responses. Okay. All right. So the first question, what are the main challenges in developing and disseminating? Oh, sorry, this is the second question. Let's go back to the first one. The main challenge you face in outlier
Errol,
detection.
sorry,
Sorry,
Errol,
Andrew?
it's Andrew here. Yeah, just a note that on my PC, the questions took a while to load. It went through some external virus checking. So I wonder if we can allow just a little while longer before you sort of close off the poll in case people have had similar issues. Yeah.
Good idea, Andrew. Yep. So I'm just going to, this is live, so I think as the responses are coming, we'll see those numbers change. But I'll wait a while until we form some firm response around, those final numbers. So keep those responses coming in. I just wanted to show you a very live version. I think the main sort of problems that are sort of creeping up, small numbers, rare outcomes. And this is really quite challenging, right? Small numbers, rare outcomes. And I'm very pleased to say that the work that Jesse has done has, basically addressed one of these as the main problems. So we do know that small numbers, rare outcomes poses a big problem, not just when we create the funnel plots, but in terms of the consistency. Like, each year when you come up with a funnel plot, they vary quite unexpectedly because of those small numbers. So I think you will find that dashboard, the interactive dashboard, really useful in terms of planning for your benchmarking exercises when you do face those problems. Now, the other interesting aspect that we see, and it's not really statistical, right? It's more about interpreting and communicating those outlier, those funnel plots that we have to a wide range of stakeholders, many of whom may not have a very grounded sort of statistical background. So I think that's one of the main challenges we do face within the registries in our school. How do you actually interpret, communicate, and then win the confidence of those who are actually reading those benchmark reports? So keep those response coming in. It's really interesting. Just in terms of other challenges in outlier detection, data gaps, I think data quality is a big issue. We'll come back to it in a minute. Delay in publication. So yes, the lag time in terms of how the data actually gets transported from the sites to the registry and then gets reported. I think this is a very valid concern. I'm just going to the next question. What are the main challenges in developing and disseminating site reports? As you can see, I mean, for many in the audience, missing data and data quality issues is one of the main concerns. Now, we didn't really cover it in the presentation that Jesse presented, but we will take note of this and then see whether future webinars we can talk about this in more detail. Automating reporting and dashboards, we have done quite a bit in this area, so we are very happy to share some of our experiences in using, in particular, R Markdown in terms of automating the dissemination of reports, as well as dashboards using R Shiny. Again, going to the, in terms of challenges not listed, we've got hospital, we've got data and risk, engagement and trust. I might just ask, whoever put the comment about hospital, if you can just maybe elaborate a bit more about what are the specific challenges in the chat box. I'll come back to that in a minute. Okay, so I'm just going to go back to stop sharing. I'll just come back to our presentation. Okay, so now it's very interesting. So thank you, Jesse. I think you covered, I think, some of the critical elements about the challenges we face in registry reporting, outliers, the benchmarking process. And I think you provided us with a solution. I think the first thing you did was to contextualize the problem. And if I may summarize, I may have got it wrong, but what you're saying is that there's no one shoe fits all size, right? We do know that the registries, they come at different stages. So it may be a small registry just setting up that's dependent on funding, and they need to produce reports. There's pressure to actually produce side reports or produce benchmarking reports, and they want to do the right thing, right, with those small numbers. And I think you're telling us that your framework actually allows them to plan confidently the key indicators, so they could select the key indicators that they can confidently present. And then you presented us the other end of the scenario where you have a large established registry, and with small departures, you sort of pick up outliers that are not clinically significant, but due to the fact that they have very large volumes, they are being flagged as an outlier. So I think you're telling us that it's a multifaceted problem, and hence your simulation study sort of gives us a framework how to actually address this multifaceted problem. So and thanks for sharing that resource with everyone. I think after the presentation, once people have had a chance to actually use the dashboard, I'm pretty sure we'll have more questions coming through to us. But at this stage, maybe I should probably stop talking and maybe ask, whether there are any specific questions anyone has on Jesse's presentation. Any specific?
Yeah, Errol, there are a few. Yeah, Errol, there are a few questions that came up in chat, and I'll just read them out.
Thanks, Andrew.
Okay, so the first one was from Ian Smith, and he said you focused in your talk just on the discrimination in the adjustment model, I think the AUC. What about the calibration of the model, like the Hodges, what's it, HL, Hodges-Lemmerschau, Hosmer-Lemmerschau statistic thing, yeah.
Yeah, so within my simulations, I only looked at the adjustment of the model. But yeah, I think that would be a very good extension of the simulation framework that or the simulation function that I developed could be to look at different calibration of models, and can also be extended to different models and different tests. Yeah.
Okay, thanks, Jesse. And please reply in the chat if you have something else you'd like to follow up on. The next question was from Jess Kaiser. Thanks for a great talk. Have you considered controlling family-wise error rates or false discovery rates? There's been some work that has been looking at this stuff, but it was quite a few years ago now. Just wondering if you could comment on that.
Yeah, so for my simulations, I did look at controlling the false discovery rates. I didn't do any, family-wise error rate controlling, but for the false discovery rates, the performance was fairly similar to I think, to the other methods that I was looking at. And so I didn't pick it as a key one to expand the simulation study on, mainly because, I picked the methods that were the most commonly used and I guess more easily interpretable by people who I was talking to. But I think, again, it would be interesting to look at them now that we've sort of, got the framework for doing that.
Okay, thanks. And Jess, of course, anything to follow up, please do in the chat. Question from Jolie Hutchinson. I have a question regarding the minimum number of sites required for meeting center level benchmarking. Whereas the guidance often refers to sort of a minimum number of cases per site rather than actual minimum number of sites, because we've only got, say, 2 sites.
Yeah. Yes.
Yeah, so what's the response comment to that?
Yeah, I used a minimum of 5 for my simulations, but again, unfortunately, yeah, I can't provide a specific answer, but I think that that would be something that's interesting to look at. Yeah, but whether, under 5 potentially isn't really useful because, yeah, there's so few to base the average on. Yeah. But yeah, that would be something that would be interesting to look at.
Yeah, that's a good question. If I may just jump in and just maybe allowed to share my experience. I remember in one of the registries that we were starting up in Monash. When you start a registry, obviously the number of registries is smaller. But as I mentioned in an earlier point that I made, I think the pressure is still constant. The pressure is constant from the funders, from the stakeholders. I mean, they want to see a report. So, and one way we sort of get around that with a small number of volume sites is actually to put some caveats around. And that's what we did for some of our initial reports. We know that they are not robust in terms of the number of sites. So we put in some caveats in terms of how you report it. But the key thing is as the registry matures, more and more sites are getting on board and you get a more robust sort of reporting mechanism. So I don't think that, we can put a number and say, look, I mean, like what Jesse said, anything less than 5 is still arbitrary, but, the pressure is intense. I mean, you do need to produce something for the registry to sustain, right? And then for them to believe in the registry and the reporting, and then to get funding, and then for you to actually do this in a long term. So I think it's a very good question. I think we need to maybe think about addressing that maybe in a future work. But at the current recommendation is if you do have a small number of sites, you have a funnel plot, you do need to put some caveats around how you interpret them. And I think that's a very balanced sort of approach.
Yeah, and I think that's the.
Go ahead, a couple of.
Oh.
Sorry, go ahead.
Oh no, just that I think that that's for me like the key takeaway is that there is always uncertainty and that this work doing benchmarking will never find outliers exactly, and it's more like what's the interpretation of it, what's the clinical significance, what are these other factors around, rather than just having a statistical equation be like, oh, this one's like statistically significant. I think communicating that to government and to stakeholders is really, really important as well.
I mean, that came out as one of the points, right? A lot of in the audience are actually, I mean, they believe in the models, but I think it's more about communicating this to the stakeholders. And maybe this is something we can tackle in a future webinar. I think we do need some good strategy about how we demystify some of these statistical exercises that we have developed. I'm sorry, I can't see the questions in the chat, Andrew, but you need to maybe.
That's fine, Errol. I'll read them out when they come in. You've answered all of them so far. Just my question, in your poll, I think the first question was about dealing with sites with low prevalence indicators. And if I understood correctly, in your analysis of the ANSCETS database, that's what the cardiothoracic surgery database, the prevalence is about 2% there. So could you sort of reconcile what one does with really low prevalence indicators?
Yeah, so for ANSCETS, I think it's actually quite a good example for that because they had 2 indicators of quite low prevalence. Mortality was around 1% and renal insufficiency was around 2%. But with the simulations of ANSCETS, their risk adjustment for renal insufficiency was really quite good. And they had relatively high case volumes for all of their sites. And so for renal insufficiency, the expected accuracy of benchmarking was, really quite high, over 80% for risk-adjusted analyses. Whereas for operative mortality, which did have slightly lower prevalence, but also the risk adjustment wasn't as good, and so the theoretical accuracy for those were quite poor for most scenarios. So yeah, I think that if possible, risk adjustment is something that's very, very important, and also waiting until you have enough cases, but that also, yeah, of course, comes with losing relevance of the data, and yeah, so it's tricky.
Okay, a couple more questions have come in. Thanks, Jesse, for that. So Claire Stewart asks, any thoughts? Is the average actually the best benchmark?
That is a great question that I would also, say would be very good to look at. Some papers have looked at the median, but I think an external benchmark would also be something very interesting to look at, especially because the average is so dependent on the data that is actually in the registry. And if you're a voluntary registry that mainly has private hospitals, your benchmark might be quite different to what the actual population benchmark is across all hospitals. And so, yeah, potentially an external benchmark could be a more appropriate measure to compare sites to.
Yeah, good question. I was starting to type some response, but usually what we observe is that, if you realize, if you take the average across all sites, the average is usually dictated by the big sites. So if you have a large sites, that contributes, quite a bit to the average that we are talking about. But really, the question we should ask is, what are we really comparing against? We're comparing against the average of all sites, and that's the default that we usually do. But what Jesse is also highlighting is that sometimes, there may be some external benchmarks, like clinical practice guidelines that establish like timeliness of care and other indicators. And if we have that certain number that's out there, I think those can be used as external sort of benchmarks. But otherwise, it's more of a statistical exercise where we're looking at where are you as a site compared to others around you. But the problem is not so much about who are the people around you. It's like, are those around you comparable? So I think that's the thing that we are sort of struggling to do. We can't like put everything into a funnel plot. We need to put, sites that are comparable. And that's the other thing I think Jesse was also looking at, like in terms of risk adjustment, is there only so much we could do in terms of risk adjustment versus, should we be stratifying sites because they are so different that, risk adjustment may not be adequate. So, these are again really quite, very difficult issues. And during it took Jesse like 3 years to study like parts of the problem. And I think there are other problems as well we haven't quite addressed. So there's probably like maybe like 2 or 3 more PhD work in this area as well, if anyone is interested.
Ian's followed up with a really good question there. The choice of the benchmark depends on what you're trying to do here. You're trying to drive excellence or drive mediocrity, I guess, putting the challenge out there. Some benchmarking uses a challenge level, the 25th percentile, to show what is achievable. So just any comment about using different percentiles there?
I think one of the things, I discovered through my role in steering committees in some of the registries is, you can have like 3 different models, and it's going to give you 3 different sets of outliers. And that's going to cause a lot of confusion and a lot of disagreement when we are trying to interpret, these funnel plots. And I think one of the main things we need to do as statisticians is to be very engaging, very collaborative. So I think there are various methods. I wouldn't say one is right and one is wrong, but the key thing is the communication. So we need to come up with a method and communicate that with the stakeholders, the clinicians, the government, stakeholders, and firm that up before we actually generate those funnel plots. So I think the 25 percentile is another method. I agree it's a good method, but the key thing we need to do is to actually do this prior to actually generating those funnel plots, get the consensus, and then we need to move forward and say, look, this is what the results are showing. There will be disagreements. Obviously, the sites that are not performing well will have problems with the risk adjustment. They'll have problems with the methodology. But if you want to do a meaningful exercise, then we need to have a structure around this. And I think Jesse's work is really crucial to allow us that first step for registry to determine what is the more sort of robust way to actually set up these outlier detection methods.
Okay. Yeah, thanks, Errol. Another question from Ian, very engaged audience participant here. Again, this follows up his previous question about discrimination versus calibration, that discrimination may be relatively consistent over time, but calibration can vary as clinical technique and technology advances. It's important to consider when building models and establishing baseline performance how the relevant historic data is to current practice. So that sort of address. Can you just make a comment about that? Do you want me to read again? It's quite a big comment, quite a long comment. Or do you got something you?
No,
Yeah.
I think that's
It's about the use of historical data when things change over time.
Definitely, yeah.
Yeah.
Can I answer? Maybe because this is a real issue. I'll give you an example. For example, the breast device registry, Australian breast device registry, when we do our benchmarking, initially we compared like devices across like 10 years. But the problem is, historically, some of those implants have been found to be problematic and they were removed from the market. So then we started revisiting this exercise and trying to look at a more contemporary time period and not compare those historical period where the devices are, were inserted but were no longer in the market. So I think this is an important point, that this, when we do the funnel plots, it needs to be on the contemporary data. We can't have a timeline that's too long and where, practices have changed, the guidelines have changed, and the data is not really comparable.
Yeah. And yeah, for the risk adjustment models, I agree. Yeah, you validate on one data set and then the patient population evolves, and then yeah, calibration is a really important consideration. So yeah, I think that that's something that would be really important to look at, especially as we now have so many more mature registries within Australia using risk adjustment models that have been validated perhaps quite a while ago. Yeah.
I've just got another sort of question about the translation of all of this. So is there an existing sort of national guidelines document for clinical quality registries reporting, and that your work will be able to contribute to? I know there's sort of a more general document. I think Monash produced about sort of CQRs and what needs to be included in a registry, more for people who are considering setting up a registry. But any comments about where this work sort of fits into the guidelines, or what sort of guidelines are out there?
Yeah. Arul might be able to better address
I can maybe
this.
help you answer that. So yeah, I think like 1 or 2 years ago, we actually we worked to come up with that CQR reporting guideline. I think that's the one you talked about, Andrew. I think we were commissioned by the department to actually come up with a guideline. Now that guideline is quite broad. So I think what Jesse is doing is a bit more micro in terms of the actual outlier detection method and in terms of providing this framework. And I think, I mean, that's the issue. I mean, there's no like one model that's universal. There's a process that we need to, maybe each registry need to sort of follow. So I think we still haven't found the right forum to maybe, I mean, this is a good start. I think I'm hoping that the webinar will actually generate some interest, but if there are some in the audience who can actually maybe work with us to actually see how we can actually raise this to the next higher platform for us to actually, see whether we can implement this in more registries across the nation. I think Jesse will be really happy to pursue that, right, Jesse?
Yes, yes. Always happy for collaboration and yeah, opportunities to integrate this. Yeah, this is a while ago, but yeah, the most recent guidelines that I've seen were very vague in regards to benchmarking. And so, yeah, I think it would be good to incorporate this work and more specific guidelines. Yeah.
Just come in. Okay, it's quite a long one. I'll just read it out. I'm reading it on the fly here. Let's say we have a risk-adjusted funnel plot that plots an outcome for a treatment year expressed as an average for each hospital. However, in the plot, there are many small hospitals with varying performance. So now sort of the trimming methods called Windsorization and control for over-dispersion have previously been investigated. And so this would lead to my sort of next question is about, which I was going to ask. So thank you, Damien, for this. Would you consider using empirical Bayes shrinkage to stabilize small hospitals and improve outlier detection? And my question was going to be generally about Bayesian methods to sort of smooth things out. Yeah.
Yeah. So for at the beginning, I decided just to use traditional methods as that's what was most common from my systematic review. But I definitely think that the incorporation of Bayes methods would be, yeah, a really good way of handling some of the issues that, yeah, traditional methods can't really help with or address. Yeah.
Yeah, I agree. I think Bayesian methods allows
generally,
us
do you think there's things that could be learned? Okay, go on, please, Errol. Sorry for interrupting. Go ahead.
Sorry, Andrew, you go ahead. I'll type my response.
No, you please go ahead. I think my internet froze for a moment. My apologies. You please go ahead, Errol.
No, I just wanted to say, I think Bayesian methods can be useful because they allow us to actually incorporate like a prior information. So I think there's a scope for us to develop a good funnel plot methodology that combines the data that we harness from the sites and then incorporate that with clinical information about what clinicians think constitutes an outlier. So I think by using Bayesian methodology, I think hopefully we can not just do the shrinkage. I think the shrinkage is important, but it allows us to combine information from different sources. So I thought that was a good, it's a sort of promising area. I think we can do more in this area.
Yeah, something Damien is suggesting there is empirical Bayes. It's something to look at. We have to wrap up in a minute. Reza has put a whole lot of things in the chat, but we're not going to have time to do that. So unfortunately, we have to wrap up. So Bernie, I hope you can save the chat here and we can provide all the questions to Errol and Jesse. If there's anything that hasn't been addressed, then maybe they can respond by email because we'll have your names attached to that. Bernie, so Bernie, do we have some closing slides to show? But in the meantime, let's thank Errol and Jesse very much for a very stimulating presentation and all for the really excellent questions that arose. And our next webinar says there'll be information coming soon. So stay tuned for that. And thank you everyone for coming along today. And again, thank you, Errol and Jesse.
Thank you, everyone. Have a good day.